Good morning. Let's start the last section of chapter 7. As a review, remember we talked about norepinephrine, serotonin, dopamine. We already talked about acetylcholine. We said a neuron that releases norepinephrine is called noradrenergic neuron. The one that releases serotonin is called serotonergic neuron. Neuron that releases dopamine is called dopaminergic neuron. And finally, if a neuron releases acetylcholine, it's called cholinergic neuron. In this column, you can find the location for those neurons that contains these neurotransmitters. Here shows the structures that this neuron will innervate, and here shows the function. We went through all of this one, and this table can summarize those information. Now, let's start a new neurotransmitter, and that is histamine. Histamine is derived from an amino acid called histidine. Majority of this neuron that contain histamine is found in hypothalamus. I showed you already the location of hypothalamus. If you remember, we said this area is called diencephalon, and this location is hypothalamus. The arrow is not pointing to the right area, it should point to this area, this hypothalamus. And this is pituitary gland. Just to let you know, this gland is being controlled by hypothalamus. Hypothalamus controls this gland also. Hypothalamus contains many nuclei, and you know what nucleus is, collection of cell body. Each of those nucleus has different function. Some of the function of hypothalamus is controlled or modified by the function of histamine. For instance, we know that hypothalamus is involved with cardiovascular control. We know hypothalamus is involved in the food intake with your satiety and hunger. Hypothalamus controls your body temperature. And as we mentioned a few minutes ago, hypothalamus controls your pituitary gland. Pituitary gland is a gland that produces some hormones. I don't want you to memorize the name of hormone right now. I just want you to know that histamine is found in the hypothalamus and controls some function of the hypothalamus. Those functions relates to cardiovascular control, like blood pressure, food intake, body temperature, and controls of pituitary gland. So for now, the name of the hormone is not super important. Let's move on to the next group of neurotransmitters, and that is amino acids. We divide amino acids in two groups, excitatory and inhibitory. Excitatory amino acid as neurotransmitter is found in the CNS, and we have two of them, glutamic acid and aspartic acid. Glutamic acid is the most common excitatory neurotransmitter in the brain. More than 50% of all neurons in the brain that have receptor for glutamic acid. Glutamic acid, majority of time, create excitation. In some cases, may cause inhibition, although, as I said, majority of time create excitation, 80% of time, but in 20% of the time may cause inhibition. We are not going to get to that inhibition function of the glutamic acid. We only discuss the excitatory action of the glutamic acid. Glutamic acid is very well known on its cognitive function, including learning and memory. In the next chapter, chapter 8, I'm going to discuss about how glutamic acid is involved in your memory. But for now, I want you to know that glutamic acid has 3 receptors, NMDA, AMPA, or it's called AMPA, and kainate. Kainate, it's very similar to AMPA, but is not as prevalent as AMPA. It does exactly the same thing. These 3 receptors are ion channels. In other words, glutamic acid is going to sit on the receptor and these channels will open. So they are ionotropic. The channel and the receptor is the same thing. NMDA and AMPA are the ones that are involved in the memory process. Glutamic acid also has other receptors that they are not channels themselves. They are metabotrophic. In other words, glutamic acid is going to sit on them and they're going to use G protein. The channel and receptor are 2 different structures. For our class, since it's a basic physiology course, we are not going to go through these metabotrophic receptors. We only discuss the ionotropic. We are going to talk about detail of these receptors in the next chapter, chapter 8, when we talk about memory. After glutamic acid sits on the receptor, like any other neurotransmitter, we have to remove them. Otherwise, the effect of glutamic acid continues. To remove glutamic acid, we're going to reuptake glutamic acid back inside the presynaptic neuron or inside the glial cells. So to remove glutamic acid, we have a reuptake system. It's called excitatory amino acid transporter, or it's called EAAT. Let me show you. In this picture, you can see here is the presynaptic neuron. This is a glial cell. Let's say that this cell is going to release glutamic acid. As you can see, this purple one are glutamic acid. When the glutamic acid is released, we have to be able to recycle glutamate. When this neuron releases glutamate, this glutamate is going to come to the synaptic cleft, and some is going to come back inside the presynaptic neuron through this EAAT and get recycled. But majority is going to enter to the glial cells, specifically astrocyte, again through the same transporter. It's called EAAT. And when this glutamic acid is going to come inside the astrocyte is going to turn glutamic acid to the glutamine thanks to glutamate synthase. Glutamine synthase needs NADPH. So NADPH is needed for this process of changing glutamic acid to glutamine. Glutamine later is going to leave this astrocyte, is going to come to synaptic cleft, and again is going to be reuptaked back inside the presynaptic neuron, and inside this presynaptic neuron, now glutamine is going to change to glutamic acid through the enzyme is called glutaminase. Here you have to correct this one. It's not GDH, but it's glutaminase. When you create glutamic acid, this glutamic acid is going to get back inside the vesicle and can be released again. So this cycle of releasing glutamic acid that comes inside the astrocyte, change to glutamine, and is going to come back inside the presynaptic neuron and change back to glutamic acid is called glutamate-glutamine cycle. There is a way to create glutamic acid, and that is through the Krebs cycle. Alpha-ketoglutaric acid can change to glutamate thanks to the enzyme is called glutamate dehydrogenase. I hope you remember something like this that I showed you when we talk about metabolism. Remember, alpha-ketoglutaric acid can change to glutamate. This is a keto acid, and by giving amino group to keto acid, you can create amino acid. Alpha-ketoglutaric acid can change to glutamate thanks to glutamate dehydrogenase. So here we can use alpha-ketoglutaric acid and turn it to glutamic acid. NMDA is one of those receptors for glutamic acid, if you remember we mentioned a few minutes ago. Ketamine, which is an anesthetic drug, is a drug that blocks NMDA. As a result, when it's used, it's going to remove aversive memory of surgery. With the same token, PCP, which is a recreational drug, is going to block NMDA and is going to impair the memory. Because glutamic acid is excitatory neurotransmitter, theoretically, if you block glutamic acid, you should also be able to stop seizure, and that is true. We have some drugs you're going to learn in pharmacology we use to stop seizure, and those are the drugs that blocks glutamic acid. Let's move on to inhibitory amino acid. The first inhibitory amino acid that we need to know is glycine. Glycine is going to sit on the receptor, which is channel also, is ionotropic, is a direct coupling. These are different names that refers to a receptor that is also a channel. When the glycine sits on the receptor is going to open up and allows chloride to move inside the cell. As you all know by now, when the chloride comes inside the cell, is a negative ion, and as a result inside the cell become negative and blocks or inhibits the neuron. We use glycine in brainstem, but more commonly in the spinal cord. In the spinal cord, glycine can regulate your skeletal system movement. For instance, if you want to flex your elbow, you have to contract your biceps and then you have to relax your triceps muscle. To do so, we need to release glycine to relax triceps muscles. In other words, you cannot contract biceps and triceps at the same time. To extend your elbow, you have to relax your biceps. The point here is this: one muscle needs to relax while the other one contracts. This relaxation happens through the role of glycine on the spinal cord. More importantly, relaxation of diaphragm, which is necessary for breathing, also depends on the function of the glycine. The poison strychnine or strychnine that is used to kill rodents works by blocking glycine receptors. By blocking glycine receptors, the respiratory muscle is going to go to contraction, and as a result, rodents be killed due to asphyxiation or lack of oxygen. The next and probably even more important inhibitory amino acid is GABA. GABA stands for gamma aminobutyric acid. It's the most common neurotransmitter in the brain. The interesting point about GABA is made from glutamic acid. Remember, glutamic acid is excitatory neurotransmitter. This glutamic acid can turn into GABA itself. The enzyme that turns glutamic acid to GABA is called glutamate decarboxylase. GABA has two different types of receptors. One is called GABA A, another one is called GABA B. GABA A is a direct coupling, or is ionotropic, or is the one that receptor and channel is the same thing. But GABA B is coupled with G protein. In other words, the receptor and the channel are two separate structures. Another term for this one is metabotropic. When GABA A is stimulated, the channels open and chloride walks inside the cell. This is very similar to the function of glycine. However, when GABA B is stimulated, the result of this one is opening the potassium channel and let the potassium leave the cell. Inside the cell becomes more negative and cause inhibition. To remove GABA, we have a GABA transporter, or you can metabolize it by GABA transaminase. By breaking down with GABA transaminase, the remaining structure can enter to the Krebs cycle. The function of GABA would be, of course, inhibitory, so therefore is involved in motor control the same way that we explained for glycine. Now, do you remember this picture we just talked about? So let's now talk about how GABA is going to come into picture. Glutamine, which is made inside the astrocyte or glial cell, can get back inside the GABAergic neuron, the neuron that produces GABA, and as you can see, glutamine can change to glutamate. Again, the enzyme is called glutaminase, but not glutamine dehydrogenase. That's a mistake. And glutamate can change to GABA, and by releasing GABA, you can inhibit other cells. GABA can come back inside the neuron through the GABA transporter 1, and inside the cell can be recycled or can be broken down by an enzyme is called GABA transaminase. When GABA is broken down by GABA transaminase, it's going to change to succinate. Do you remember succinate is also part of the Krebs cycle? Do you remember Krebs cycle? When you create your succinate, this succinate also can turn into alpha-ketoglutaric acid, and we said alpha-ketoglutaric acid can turn into glutamate thanks to glutamate dehydrogenase. So now let's take a look at the whole cycle one more time. We mentioned that glutamate is going to get released inside the presynaptic neuron. Some is going to be reuptaked back inside this neuron, but majority is going to come inside the astrocyte. Astrocyte is going to take this glutamic acid in and turn it to glutamine. Glutamine will be released inside the cleft and can come back inside the presynaptic neuron. Inside the presynaptic neuron, we can change glutamine to glutamic acid and recycle it. We also said we can create glutamic acid from alpha-ketoglutaric acid. We also mentioned that GABA is made through the glutamic acid. Glutamic acid can create GABA. When the GABA is released inside the presynaptic neuron, after it does what it's supposed to do, can be recycled back inside the cell or can be broken down by GABA transaminase. GABA transaminase is going to turn GABA into succinate. We said succinate can go through the cycle of Krebs and create alpha-ketoglutaric acid again. And alpha-ketoglutaric acid then is going to turn back into glutamic acid, and this cycle can goes on and on. And for the last time, this slide shows what we just mentioned. Glutamate is going to change to gamma aminobutyric acid or GABA thanks to glutamate decarboxylase. GABA, after being released, can be reuptake inside the presynaptic neuron to be recycled, or you can break it down by GABA transaminase and change it to succinate. And succinate coenzyme is part of the Krebs cycle and can turn to alpha ketoglutaric acid, and alpha ketoglutaric acid can turn back and create glutamate again. So now let's talk about a couple of clinical vignettes for this one, so make sure you understand how important this information is. GABA A, that we said is linked to the chloride channel, is a target for many drugs that we use as sedative. For instance, benzodiazepine. People, they take it before going to surgery. People, they take it when they are stressed out or they want to sleep. Those drugs works on GABA A. It's going to open GABA A and it's going to bring chloride inside the cell and slow down your neurons' activity. With the same token, barbiturate also works based on the GABA A. So by learning this one, you're literally improving your understanding of pharmacology also. Also, I want to talk to you about Huntington. There is a disease called Huntington disease. In the Huntington disease, people, they're going to lose or degenerate their GABAergic neurons, the neurons that release GABA as a neurotransmitter. They start moving unintentionally. This type of movement is called choreiform movement. These people, they have hyperkinetic, they move too much. And this choreiform movement is due to lack of the neuron that releases GABA. As you know, GABA is inhibitory, so they lose this inhibition and start moving without intention. GABAergic neurons, the neurons that release GABA, are super important to control your muscle activity. This picture shows GABA A. As you can see, we need 2 GABA to open this channel. As soon as 2 GABA sits here, channel is going to open and chloride is going to come in. So to open GABA A, we need 2 GABA to be able to open the chloride channel. However, GABA B, as you can see in this picture, GABA B, the receptor and the channel are different. Here's the receptor and here's the channel. When the GABA B is being stimulated, as a result, opens potassium channel. Potassium is going to leave the cell and this cell becomes hyperpolarized. Now let's move on and get to the polypeptides. Polypeptide also can be neurotransmitter. Those polypeptides that act like a neurotransmitter are called neuropeptides. Many of these neuropeptides that are released inside the brain from neuron, also they act as hormone. For instance, CCK is a hormone released from gland. So these are the chemicals, not only they act as hormone and they release from the glands, but also they act as neurotransmitter and they release from the neurons. Example for this one would be CCK. CCK is a hormone, but also is a neuropeptide. Substance P can be also a chemical that released from regular cells, but then also is released from neurons. The first neuropeptide that I want you to know is called substance P. P stands for pain. It's a mediator for pain signal. When you feel pain, majority of time is because of releasing this substance from the cells or from the neurons. Beta endorphin, dynorphin, or enkephalin are also neuropeptide. They are released from neurons and they are opioid. They are similar to opium. They do the same function, but you have it in your neurons. By releasing this opioid from the neurons, of course, the patient is going to feel euphoria, feels happy, relaxed. When you exercise, when you eat and you're relaxing, your body is going to release opioid. So as a result, you feel good about yourself. Opioid or opium, they have so many great functions. That's why your body has it. Obviously, abusing it is going to cause trouble. But in the normal amount, when your body produces it, it's going to give you the sense of euphoria, decrease pain, is going to adjust your blood sugar, and has very important role in homeostasis. These opioids are produced inside the brain and also inside the pituitary glands. They use second messenger to function. In other words, the receptor and channel, they are not the same thing. It's metabotrophic. The next neuropeptide is called neuropeptide Y or NPY. Neuropeptide Y deals with appetite. Neuropeptide Y is the most common neuropeptide in the brain. It's synthesized and released in the hypothalamus. Remember, hypothalamus is responsible for satiety and sense of hunger. Neuropeptide Y is released from hypothalamus and stimulates the sense of hunger. I don't know if you heard about leptin. If you go to Whole Food and look at those bottle of the pills that claim that is going to stop your appetite, you can see one of the active ingredient of those bottles are leptin. Leptin is a product that inhibits neuropeptide Y. Remember, neuropeptide Y is going to increase or stimulate your hunger, but leptin is going to block it. There are two centers in the hypothalamus, or there are two nucleus in the hypothalamus. One is called the ventromedial nucleus, another one is called lateral zone. Ventromedial nucleus is a target for leptin. Leptin is going to stimulate this nucleus, and by stimulating this nucleus, you're going to feel full. On the other hand, leptin is going to come to the lateral zone and is going to inhibit lateral zone, because lateral zone is a center in your hypothalamus that makes you to feel you're hungry. And leptin is going to come to this guy and block it. So leptin by stimulating ventromedial is going to give you a sense of fullness, and by blocking lateral zone is going to make you to feel less hungry. And all this function is going to happen by inhibiting neuropeptide Y. Another function that probably you need to know for neuropeptide Y, besides its function on appetite, would be the role of neuropeptide 1 on stress, circadian rhythm, and cardiovascular. Since neuropeptide Y is synthesized and released from hypothalamus, it seems like affect other function of hypothalamus. We mentioned a few minutes ago that hypothalamus has so many functions. Now I want you to know that hypothalamus is responsible for your emotion. It's going to deal with your stress. Hypothalamus also is responsible for your circadian rhythm, when you wake up and you sleep, and cardiovascular control. And lastly, neuropeptide Y is shown that inhibits the release of glutamate in hippocampus. Hippocampus is part of your brain that deals with memory. And we said a few minutes ago that glutamic acid is the most common excitatory neurotransmitter in the brain. It seems like neuropeptide Y also inhibits the release of glutamate in the hippocampus. That is going to bring us to the gut-brain peptide, because not only they are found in the brain and neuron, but also they are found inside the GI system, like cholecystokinin or CCK, or somatostatin or VIP. We're going to talk about these chemicals in the GI system as well. So for now, CCK is a hormone and also neuropeptide that deals with feeling of satiety. When you increase inside the brain, it's going to make you to eat less. Somatostatin and VIP are other gut-brain peptide that we talk about those two later in the GI system.